Welcome, everyone. My name is Jaymar. I am a senior marketing associate here at Nanion Technologies, and I'm happy to be your host for this webinar today. So first of all, thank you for joining us, as we will be exploring on channelopathies and lysosomal targets with the SyncroPatch. Today we have two featured talks. We will start with one by Dr. Ivy Dick from the University of Maryland. And the second talk is by Ewan Rees Williams from Cardiff University. Okay, so to get started, I would like to then introduce my colleague, Ali Obergrusberger. She is the Director of Scientific Sales and Customer Engagement here at Nanion Technologies. She will start the webinar by giving you some information, and she has also an announcement for you all. And later she will be moderating also the Q&A session. So do not hesitate to write your questions, and it is then my pleasure to leave you with Ali. So Ali, whenever you're ready, you can take over.
Thanks, Jaymar, and good afternoon, good morning, wherever you are. Welcome from me also to this webinar this afternoon or today, exploring channelopathies and lysosomal targets with the SyncroPatch 384. Before I introduce the speakers of today's webinar, I just wanted to sort of remind everyone who is listening of Nanion's commitment to supporting your research. So academic research is really in our DNA, as Nanion started off as a spin-off from LMU here in Munich over 20 years ago. In addition, most, if not all, of our application scientists, and indeed much of our sales and marketing team, have a background in academic research and are MSc or PhD level graduates. At Nanion, we regularly participate in PhD programs, and we love to collaborate with researchers to generate data for joint manuscripts for publication. We also participate in teaching workshops such as the Cell Physiology Workshop, formerly in Plymouth, now in Liverpool, the Montpellier School of Electrophysiology, and the Young Physiology Symposium in Münster. And we also foster academic research with travel bursaries and, importantly, our instrument grants, which we've been running since 2018 for the SURFA, FlexSite, and the SyncroPatch 384. Now, we first ran the SyncroPatch 384 instrument grant in 2020 and chose two winners, the first being Angelika Lampert and Ralph Hausmann at the RWTH Aachen, with their proposal, Sodium Channelopathies as a stepping stone for personalized clinical pain therapies, and Fernanda Lieza and team at the University of Texas with their proposal, Automated Planar Electrode Electrophysiology for probing function and pharmacology of ion channels. Their research was highly successful with the SyncroPatch 384, with several publications and conference presentations resulting from the 9-month grant period. In 2023, we ran the SyncroPatch 384 instrument grant again, and again chose two winning labs to receive the SyncroPatch 384 for 9 months. These were Ivy Dick and Matt Trudeau from the University of Maryland School of Medicine, with their proposal evaluating calcium channelopathies across multiple cell types, and the team of John Atack, Helen Waller-Evans, Emir Lloyd-Evans, and Owana Popa for their proposal enabling hit identification and early structure activity generation for novel ion channel targets. And it's from these labs we're really delighted to hear from today. But before I introduce our first speaker, I do have an exciting announcement to make, which Jaymar just mentioned, and that is that we are going to run the SyncroPatch 384 instrument grant again this year. So again, we're giving a lab the chance to win a SyncroPatch 384 for 9 months, along with consumables and technical and electrophysiology support. So you've basically got the summer to think about how you would like to use a high throughput automated patch clamp device in your lab. You can register your interest on our website or scan the QR code, which is currently on this slide. And you'll be notified then when applications are open, which will be in September, and they'll be closing in October. So we're very much looking forward to finding out more about your research and to choose a winning lab to receive the SyncroPatch 384. So without further ado, we were going to hear now from our two grant winners from 2023. They received the instruments in Q1 of 2024. So first of all, we're going to hear from Ivy Dick, and then we'll move over to Ewan Rees-Williams. And after that, we're going to have a Q&A session. So I hope you'll stick around for that. I do encourage you, as Jamar mentioned, to write your questions into the Q&A box during the talks, and we can direct the questions to the relevant person at the end of the talks. So our first speaker today is Dr. Ivy Dick from the University of Maryland School of Medicine. Ivy began her studies of ion channels at Merck at the laboratories of Charlie Cohen and Owen McManus, after which she joined Johns Hopkins University as a graduate student in David Yu's lab, where she stayed on as a postdoctoral researcher after she graduated, and then as a research associate. Ivy's research focused on understanding the spatial selectivity of calmodulin regulation of voltage-gated calcium channels and how those mechanisms are disrupted in Timothy syndrome. Ivy is now an associate professor at the University of Maryland, where she continues to study calcium channelopathies.
Well, thank you so much for that wonderful introduction and for inviting me to speak today. So I'm going to talk to you a little bit about some of the things that our lab has been doing and how the SyncroPatch is feeding into those studies. So our lab studies voltage-gated calcium channels, and we've been particularly interested in mutations in CAV 1.2, which are known to cause a disease called Timothy syndrome, also called long QT type 8. So Timothy syndrome is a multi-system disorder characterized by craniofacial defects, syndactyly or webbing of the fingers and toes, and severe long QT syndrome, which is often fatal in early childhood. The underlying cause of Timothy syndrome was originally described as a single point mutation within the CAV 1.2 channel. And since that first description, the number and variety of mutations within this channel have grown tremendously. And so just looking at this, you can tell already how a high throughput method like the SyncroPatch is really going to help with a lot of these studies and moving forward. So what I'm going to do is I'm going to first describe the stories, tell you a little bit about how manual patch has led to some of our hypotheses, and then show you how the SyncroPatch that we've had in the lab for the last year has really helped us. So to begin, we looked at the canonical Timothy syndrome mutation, so the first mutations that were described. And one of the first things that was found was that these mutations affect voltage-dependent inactivation, or VDI. So for wild-type channels, we can see VDI as the decay in a barium current as it crosses the channel, and we've highlighted it in blue here just for reference. And what we found is that both Timothy syndrome mutations almost completely abolish voltage-dependent inactivation. If we look at calcium-dependent inactivation, this can be seen as the stronger decay of the calcium current as compared to the barium current. So we've highlighted that in pink here for reference. And what we found is that the initial Timothy syndrome mutations also significantly blunted this. So they're both sort of gain of function type effects on the inactivation of the channel. However, given the location of the mutations on the distal S6 region, we expected effects on channel activation. And so looking at the activation curve as a function of voltage, what we found is that even for these first two canonical Timothy syndrome mutations, the G402S had a right shift in channel activation, while the G406R had an opposing left shift in channel activation. So we're starting to get a little bit more of the diversity going on with these different mutations, where the deficits in inactivation have relatively consistently been deficits in one form or the other, but the changes in activation are a lot more variable. And so we've pursued this quite a bit, and we've looked at a number of different mutations, and you can see this pattern sort of bearing out where some mutations have more selective effects on one form of inactivation or the other, and a lot more variable effects on channel activation. And so what we really wanted to know was, does this tell us anything about the mechanism of pathogenesis in the patients or the phenotypes that are being displayed? And so if you put the phenotype for these patients, the major phenotypes that have been described, you start to notice a pattern. And all of these mutations that I'm showing you here have that severe long QT phenotype, the severe cardiac phenotype, and they all have a deficit in channel inactivation, either VDI or CDI. And we think that that's important. We think that for the heart, these inactivation deficits are really driving the pathogenesis. To look at that, we did use iPSC-derived cardiomyocytes. So we have cardiomyocytes from a Timothy syndrome patient that were generously provided by Dr. Masayuki Izawa, and we have a number of the other mutations now CRISPRed into the same background of the cells. And with that, we're able to look at the biophysical profile of these cells. Now, since these mutations are now patient mutations within iPS cells, they're heterozygous in nature. For the 406, they're actually only in a single splice variant. So we don't expect the effect to be quite as severe, yet we do see that strong deficit in channel inactivation with the 406. One of the other mutations that we looked at, we have a change in inactivation here that matches the hex cell experiments as well. Likewise, the left shift in channel activation remains for both of these mutations, although to a smaller magnitude as expected for heterozygous mutation. What this means for the action potential, so we current clamp the cells and we can look at the action potential, look at the restitution curves as well. And what we found is that for the very severe 406R mutation, you get these really prolonged action potentials that are consistent with that severe long QT phenotype. And if we look at some of these other mutations, this is a mutation that has a more modest effect on channel inactivation, but a really, really large, a much larger effect on channel activation. And it seems like that long QT phenotype, while it's still there, it's kind of following the magnitude of the inactivation deficits. Likewise, a mutation that has just a selective effect on channel on CDI also has that prolongation of the action potential. So it seems like inactivation is really important in the heart, and calcium-dependent inactivation in particular is the major driver of the long QT phenotype. Just to show what that means for the patient as far as producing some of these arrhythmias, this is an optical mapping setup, so we're stimulating right at the corner here and watching the action potential as it propagates. So the optically recorded action potential is here. If we do this for the G406R or the 1166T or 1475, what you see is that you get these episodes of spiral waves. So it's indicative of a reentrant arrhythmia. And you get this regardless of which mutation, even that small effect on CDI. So we really do think that in the heart, calcium-dependent inactivation is of primary importance in setting that pathogenic long QT phenotype, with voltage-dependent inactivation following just behind, while activation plays a smaller role in that pathogenic mechanism. Looking at the neurological phenotype, so what you notice from the mutations I showed you is only some of the mutations cause that neurodevelopmental delay phenotype. But those are the mutations that have the left shift in channel activation. And so we started off by asking whether or not that left shift in channel activation in a hex cell, so where you don't have all the downstream signaling and all the feedback regulation of a neuron, whether that left shift is sufficient to explain a difference between the mutations. So what we did is we applied a neurological stimulus, so a train of action potentials, and we recorded the calcium current in response. And you can see the peak over time during the train here. And if you do that for one of the mutations that has that significant left shift, you can see that compared to wild type, there's a significant difference in the result. Likewise, for the canonical G406R, there's a difference between the wild type and the mutant behavior. So both of these have the left shift. But all of the long QT selective mutations, the mutations that don't have as much of the severe neurological phenotype, we found that they really don't differ between the mutant and the wild type behavior. And Likewise, if you actually measure the half-width of the action potential response to the calcium response to the action potential, you find that only the two mutations with that left shift cause a change in the profile of the action potential. Since this was HEX cells, we also looked at this in neurons, and we can see the difference in the inactivation profile. And what we found, these are iPSC-derived neurons that are using the NGN2 protocol, so excitatory-like neurons. And what we found is that we have two functionally distinct populations of cells, ones that fire repeatedly and ones that fire only a single action potential, no matter how much of a stimulus we give them. And this occurs with the Timothy syndrome cells as well. However, the firing rate within the burst firing, within the repetitive firing, is higher. And the number of cells within this population is also higher. And so you're starting to see why, for manual patch, we're sort of outgrowing the ability to do this just with conventional patch methods, because we really do have to look at these population level effects where the population itself is changing and the effect of the mutation is actually quite different within one population versus the other. But the bottom line is that we think for the brain that these left shifts in channel activation are really driving that severe neurological phenotype. Not to say that inactivation isn't important. We actually do believe that it has a significant role, but that severe neurodevelopmental delay that's described in Timothy syndrome, we do think correlates with a significant left shift in channel activation. And then finally, we wanted to know whether or not this could give us any insight into some of the therapeutics for the patients. When the mutation was first described to be a mutation in the calcium channel, particularly mostly a gain-of-function mutation, it was thought that drugs like verapamil, calcium channel blockers, should work. They should really help the patients. But the reality is that most patients still rely on those internal cardiac defibrillators. And so we looked a little bit as to why that's happening. And what we know about verapamil is that it binds preferentially to the open and inactivated state of the channel. What that means is that if you give a step depolarization, you get this accumulation of block during the opening and inactivation of the channel. And that can be seen by measuring two different dose response curves. So if you measure the curve right at the start of the depolarization, so right after the channel was closed, you get one dose response curve, and then you get this sort of activity dependent shift. However, for the Timothy syndrome mutations, they don't like to inactivate. And so you actually lose some of this activity dependent shift. And for the mutation that also doesn't really like to open, you almost completely lose this. And so that means that you're going to hit the wild type channel more than you hit the Timothy syndrome channel if you treat patients with these drugs. So that really reduces the therapeutic. And so we wanted to continue to explore this and to look at more mutations, to look at these different mechanisms. But as you can see, the number and the variety of mutations is growing, the number of mechanisms we're looking at, the number of biophysical properties. And so that's where the SyncroPatch really can become quite enabling. And so we had the luck of winning the instrumentation award from Nanion, and we've had the SyncroPatch in our lab for about a year now. And we've been able to look at these same questions using this instrument. And one of the first things that we wanted to look at was some of the more recent mutations actually don't have much reported cardiac effects. So if we're correct, if we believe that left shift in channel activation is really important for the neurological symptoms, then we should be able to see that in some of these mutations. So these are mutations that produce neurodevelopmental delay. They produce seizures, but in the literature, there isn't much reported effect on the heart. Now, one of the issues that we have moving to the SyncroPatch, so commonly these instruments are used with cell lines. However, we have numerous mutations in the channel, and we don't want to have to make a cell line for every one of these mutations. So what we really wanted to do was to transfect the cells with the calcium channel. And so that's easy enough to do. but the calcium channel comes with various subunits. And so the beta 2A or any beta subunit is required to be able to look at the channel. Alpha 2 delta is required. And then calmodulin is the calcium sensor. And so in order to look in bulk at sort of the VDI versus the CDI effect, we wanted to be able to modulate CDI to remove it by using some mutant calmodulins. So that gives us a minimum of 4 plasmids that we need to be able to consistently transfect. When we started this, we actually had 5 plasmids. And what we found was that we were actually able to do this even without having to resort to sorting the cells by using the MaxCyte system. And so we're able to somewhat reliably get 4 different plasmids expressing at high enough levels to put the cells onto the SyncroPatch. So with that, so all the data I'm showing at this point is recorded from the SyncroPatch. And one of the first things you notice is it looks a lot like the conventional patch clamp data. So it gives us very nice results, very consistent with what we've gotten before. So this is barium data looking at voltage-dependent inactivation. And one of the things that we noticed is that unlike the other mutations, voltage-dependent inactivation is completely normal in these cells. So that kind of makes sense since we're hypothesizing that this is going to be mostly a change in channel activation to produce those neurological phenotypes. We next looked at calcium-dependent inactivation. So we switched the protocol here a little bit. So this is a cell with wild-type calmodulin co-transfected that's permissive of calcium-dependent inactivation versus cells where the CAM is mutated so that it cannot bind calcium. And so that gives us our control for voltage-dependent effects. And you can see that in the population data here. So this U-shaped dependence on voltage for CDI is what we expect. So this is all consistent with conventional patch clamp, confirming that the SyncroPatch is giving us very, very similar results. And what we found with the neurological mutations is that CDI is completely spared. In fact, if you look at the more negative voltages, CDI is significantly more. And so the kinetics are changing slightly, but in the 300 millisecond action potential, you would expect accumulation of inactivation. And so this could actually explain why these patients don't have a significant cardiac effect. They don't have a deficit in inactivation. If anything, they have an increase in inactivation, which might kind of be protective of the heart for any other effects that these mutations are impacting. Looking at channel activation, now here is where this is a tail current protocol that we applied on the SyncroPatch. And this is where we believe that there should be a left shift in channel activation if our hypothesis is correct. And in fact, we see for both mutations a significant left shift in channel activation. And so we fairly confident that this mechanism that we hypothesized here seems to be what's going on, that we have these left shifts in activation in the neurons that's causing that severe neurodevelopmental delay. And then finally, I showed you with the conventional patch clamp that a lot of these inactivation changes can change how calcium channel blockers are working. Well, now we have an inactivation change that it kind of slows the inactivation, but it makes it more complete. And so we think that that's gonna affect the verapamil binding to the channel, but actually maybe in the other direction. So if you look at wild type channels, so I'm just showing exemplars because we didn't fully get this analyzed before the webinar, but adding verapamil blocks the wild type channel as you would expect, but it blocks the mutant more. So now we have a situation where instead of being a detriment to treatment, these changes in activation actually might help increase the efficacy of some of these calcium channel blockers. Going forward, we want to use the iPS cells, the cardiomyocytes and neurons, as I showed you before. And so this is just to show these are iPSC-derived cardiomyocytes on the SyncroPatch. And this is just an exemplar. This is one of the most severe mutations. It's expressed at higher levels The TS2 phenotype is in a splice variant that's expressed very highly, and you can clearly see the difference in inactivation on the current. And if you look at the action potentials, so we can get nice action potentials. And this is the same mutation, but expressed at a slightly lower level. That's the Timothy syndrome type 1 expression level. And you get that prolongation of the action potential. So the SyncroPatch is picking up the same thing that we were able to see with the manual patch clamp. Looking at neurons, so these are iPSC-derived neurons on the SyncroPatch, and you can get the nice current recordings and voltage clamps, so sodium and potassium. The calcium current kind of gets swamped here, but if you use solutions that isolate the calcium current, then you can see a nice calcium trace. And then if we can look at the current clamp using these same cells, what we find is exactly the same thing that I showed you before using manual patch, where we have these two different populations where some fire repetitively and some give a nice single action potential. And so we're getting very, very consistent results with the SyncroPatch compared to what we got before with manual patch, except now we can do it in a 384 format, which really helps with all of the different mutations and the different studies that we want to do. And so overall, what we've been able to use the SyncroPatch for is to really define that left shift hypothesis, to show that mutations that correlate with more of a neurological phenotype really do have that left shift in channel activation. And interestingly, these biophysical changes that can be a challenge for treating some patients, depending on the mutation itself, actually can be an opportunity for treatment, where if the inactivation changes go the other way, it actually improves the drug efficacy. And so we think the SyncroPatch 384 is going to really enable us to look at a lot of these mechanistic changes in various mutations across various channels, and to really parse out some of these phenotype biophysical effects. And then before I end, I just want to show a little bit. So our lab does a lot of calcium channel work. These are not the only cell types that we used on the SyncroPatch. So we've done multiple calcium channels. CAV 2.1 is shown here. This is the SyncroPatch data. We've done CAV 1.3, and these are all transfected, and they work quite well. One of our collaborators has done NMDA. Again, this is transfected cells, hex cells with the plasmids transfected. Likewise with hERG, we can do this in transfected conditions. We can do a couple different cell types. So we've tried iPSC-derived smooth muscle cells and looked at their calcium currents, and you can see a nice, it's small, but it is a nice current here. And we know that it's got a significant amount of L-type current, because when we add BK, we get a nice potentiation of current. We can do this with hippocampal neurons. So these are neonatal rat hippocampal neurons that we can get nice currents and current clamp recordings from. And likewise, adult ventricular myocytes from mouse. We're able to get nice sodium currents, nice calcium currents, and I don't have the action potential data, but that should be there as well. And so with that, I'd like to thank the members of my lab who actually did this work. Kevin has really spearheaded a lot of the SyncroPatch work and has been instrumental in getting pretty much all of the data that I've shown you. I don't have Tim on here from Nanion. I should. He has been really very, very helpful in getting things up and running, getting things going. And of course, I'd like to thank our support, both from NIH, Maryland Stem Cell, and from Nanion for the instrument that we found to be really instrumental in getting our work moving forward. And so with that, I will pass it back to Ali.
Yeah, wow. Thank you so much, Ivy. This is amazing. And certainly it looks like you've been putting the SyncroPatch definitely through its paces. So thank you so much. So we're going to move on to our second speaker now. Our second speaker is Ewan Rees-Williams. He is a PhD student at the Medicines Discovery Institute at the University of Cardiff. Ewan did his undergraduate degree in biology at the University of Bath in the UK, focusing on cell and molecular biology. and neuroscience, after which he did a couple of jobs in industry before starting his PhD in 2022 at Cardiff. Ewan was using the Porter patch and patch liner when he first started his PhD and has been lucky enough to accelerate his PhD research project using the SyncroPatch since they got it in 2024. Ewan, a native Welsh speaker, is going to talk to us about lysosomal targets and drug discovery.
Thank you very much. I think accelerating data generation in my PhD project is even an understatement. The amount of throughput we've been able to get from the SyncroPatch. So I want to start off, obviously, say thank you to Ali for inviting me and for that lovely introduction. And to Ivy as well. Really enjoyed your talk. It's really nice to see what can be done with the SyncroPatch outside of the drug discovery space as well. And I really enjoyed those videos with the action potentials as well. They were great. So I'm going to be focusing more on what the SyncroPatch has done to enable us in early stage drug discovery at the Medicines Discovery Institute. I'm going to talk about our work into lysosomal ion channel targets, but I'm mainly going to focus on our ongoing TMM175 project. I can, oh, there we are. So the Medicines Discovery Institute is an industry standard drug discovery institute based here at Cardiff University. We aim to translate biomedical research into drug discovery projects, leading right the way through to clinical trials and to develop drugs for unmet clinical needs. We have an expert leadership group and a diverse team of biologists and chemists from a range of disciplines who all work together to achieve this aim. The Lloyd Evans Lab has recently joined the Medicines Discovery Institute and has been studying lysosomal proteins for the last 15 years. The lysosome is a specialized organelle which is responsible for degrading cellular waste during the process of autophagy. One of the key characteristics of the lysosome is its acidic lumen, which enables its resident acid hydrolases to break down stored waste components. Historically, the lab has worked on phenotyping individual lysosomal storage disorders, which are often characterized by defects in lysosomal proteins, which have led to insights into potential therapeutic approaches. Most recently, work in the lab has aided the transition of a therapeutic for one lysosomal storage disorder into phase 3 trials. So as Ali said, in 2024, Cardiff and us at the Medicines Discovery Institute won the NANI and SyncroPatch grant, which provided us 9 months of in-house use to aid the Medicines Discovery Institute's ongoing drug discovery projects. The grant came with in-person training, ongoing support, as well as consumables for the 9-month period. Following the success of this 9-month period, we subsequently won a grant to purchase the system through the Welsh Government. The SyncroPatch itself is a high throughput, as we've obviously just been described, is a high throughput automated patch clamp system, which allows electrophysiological measurements in a 384 well format. The SyncroPatch has been pretty fundamental in the progress so far of our TMM175 project, which I'll be describing for the rest of the talk. So TMM175 is a component of the lysosome's pH homeostasis system. Defects in lysosomal pH are prevalent in lysosomal storage disorders, as well as more prevalent neurodegenerative diseases, as well as being a hallmark of the aging process. Lysosomal pH is tightly regulated by VATPAs, counter ion fluxes, and the proton leak channel TMM175. So VATPAs pumps protons into the lysosome, utilizing cytoplasmic ATP. Counter ion fluxes maintain the lysosomal membrane potential during VATPA's function. And finally, TMM175 is a proton leak channel, which counterbalances the import of protons and potentially sets the pH set point of the lysosome. We believe that TMM175 modulation could fine-tune lysosomal pH and is a more attractive target for from a drug discovery perspective than BATPAs for lysosomal pH modulation. TMO175 loss of function mutations are also associated with Parkinson's disease, meaning that TMO175 modulation is also a potential therapeutic avenue that is currently being explored for PD. So our project began with the generation of a stable cell line, a HEC cell line expressing flag tag TMO175. This cell line was then subsequently validated using Western blot and immunostaining, which we can see here. This was done with both anti-flag and anti-TMO175 antibodies. Upon receiving the SyncroPatch, having done some preliminary work with the patch liner, we then validated that we could measure TMO175 characteristic currents on the plasma membrane of this cell line. This involved the measurement of current activated by zinc, as well as being inhibited by 4-aminopyridine, which are activators and inhibitors, respectively, of TMO175. And successful validation of the cell line led to us beginning the development of an assay for TMO175 modulators. So the general principle for the assay is the measurement of TMO175 dependent current throughout solution exchange periods, which include test compounds and end with a full block with 4-aminopyridine to determine the baseline TMO175 current. Each point on the trace here represents a data point extracted from a single IV ramp, which can be seen on the right. These are repeated every 10 seconds throughout the whole solution exchange protocol. We encountered several challenges during the development of our assay. Overcoming these challenges was aided a lot by the SyncroPatch's low throughput mode, where we measured as little as 32 wells of the 384 on each chip. And this allowed us to run our optimization very resource effective and allowed us to find optimal conditions without burning through our allocation of chips from the grant. The first challenge we encountered was measuring currents of sufficient size on the plasma membrane. This challenge is not uncommon amongst lysosomal ion channels, as they predominantly belong in the lysosome, but is exacerbated slightly with TMO175, as there are no known lysosomal targeting motifs, which could otherwise be mutated, leading to plasma membrane targeting with some other lysosomal ion channels. Initially, we tested the use of inhibitors of endocytosis, a technique which was published a few years ago, specifically with TMO175. As TMO175 seems to translocate to the plasma membrane naturally through a cyclical process, it was thought that inhibiting endocytosis would result in a buildup of TMO175 on the plasma membrane, which could then be measured. And we did find that overnight treatment with these endocytosis inhibitors was able to increase the amount of 4-AP sensitive current that we could measure on the plasma membrane. But despite this success, we did decide to also try and explore other avenues to increase our plasma membrane expression, mainly due to concerns with convoluting our primary screening assay with additional compounds. We did subsequently find that changing the culturing conditions that we kept the cells under could increase the 4-AP sensitive current on the plasma membrane twofold. While this is more modest than what we got with the addition of the endocytosis inhibitors, we determined that this would provide a cleaner screening assay with less variables to worry about. The second challenge we encountered was fairly TMO175 specific, and this was establishing robust quality control parameters for the assay. as TMM175 is a leak-like channel, it is active across the range of voltages that we test. So -80 to 80, it's active throughout most of that range. So for the determination of leak seal resistance and leak currents, we determined initially that the best range to run our test pulses would be in the negative range, so it's like highlighted here. However, when we began compound testing, we found that this became convoluted. The change of the reversal potential with TMM175 modulators meant that we weren't able to accurately test pulse for seal resistances and leak currents anywhere along the IV ramp. And this meant that we moved away from traditional parameters used to assess the quality of patch clamp methods, being seal resistance and the determination of leak current, and we moved more towards ensuring that the 4AP control adhered well to what we would expect, which would be a reduction of total current to near zero. The third challenge, was also a consequence of TMM175's leak-like properties. And this was the challenge to find a balance between signal amplitude on one hand and the fidelity of the signal. So initially we determined for the traces, we took data from the 80 millivolt section of the ramp with the idea that a higher millivolt would mean we get more signal, more current to inhibit or activate. However, we found that the application of 4AP under these conditions failed to inhibit currents to zero, indicating that we had non-TMM175 specific leak currents, and these were variable between cells. So we instead moved to testing at 0 millivolts, and we found that in this range, the leak current was minimal due to the lack of an electrical gradient, which resulted in reduced overall currents. But we found that what remained was almost entirely 4AP sensitive, indicating that there was a larger proportion of TMM175 mediated current over general leak current from the seals. The fourth challenge that we had to overcome was optimizing culture conditions with some specific issues with the phenotypes of the TMM175 expressing HEC line. So we can see in the images here that unlike the parental cell line, the TMM175 expressing cell lines seem to favor cell-to-cell contacts, meaning that these cells form clusters and clumps and doing usual culturing routines and subculturing. And this made the preparation of a single cell suspension, which is ideal for good success rates with the automated patch clamping, much more difficult. As you can see here on the right-hand side, after a simple enzymatic treatment, the parental cell line is a mixture of single cells and some small clusters, which were then easily separated following manual trituration. But we found that the TMM175 expressing cell lines remained in these large clusters following being put into suspension through enzymatic treatment. To overcome this issue, we noticed that plating the cells onto poly-L-coated plates resulted in a promotion of cell to basal adhesion and resulted in much fewer cluster formations and gave us the ability to harvest high-quality single-cell suspensions. And this vastly improved our success rates. So overcoming all of these challenges has resulted in the development of a robust assay that can measure both TMM175 activators and inhibitors. So you can see here when DMSO is applied as the compound, we get minimal responses, and we get good solid activator and inhibitor responses. So validating the assay, so over the last 6 runs that we've ran, we've got an average Z prime factor of around 0.6, which for a cell-based assay is considered pretty good, as well as individual well success rates of almost 90%, meaning that we average around 340 successful wells per chip, meaning the amount of usable data we get is extraordinary from each chip. And we also found that we get around 2 nanoamps of current per cell on average, which gives us plenty of room to inhibit and activate these currents to test modulators. So the establishment of the assay has subsequently allowed us to conduct screening and a structure-activity relationship campaign, which is currently ongoing with our TM175 modulators. This layout here represents a typical plate layout for single point testing. With 8 apertures per well, a single assay can measure currents from up to 3,000 cells, which is an unprecedented throughput for patch clamp electrophysiology. This also allows us to assess up to 80 compounds per plate, and I'm able to test up to 320 compounds per day at single point. And this is partially enabled by the possibility of conducting onboard dilutions and onboard compound plate setups with the liquid handling robot, which is a part of the SyncroPatch system. And here we have some example data from a recent plate that I've ran, and here the same data where the hits have been highlighted based on deviation from the DMSO control. We've also been able to use this assay to generate concentration response curves of tool compounds and novel TM175 modulators to both validate the assay and to better characterize and compare our hit and lead compounds. The SyncroPatch enables us to generate up to 6 to 12 CS concentration response curves at 8 points per plate, which results in 24 to 48 per day. Typically 4 to 8 wells are assessed per compound, with 32 to 64 cells per compound, per concentration. So here we have an example of a CRC of the tool inhibitor 4-aminopyridine, which comes out with an EC50 value of around 30 micromolar, which aligns nicely to previously published values in the literature. We've also successfully generated CRCs for tool activators such as zinc and DCPIB, as well as novel TM175 modulators using this system. This has provided valuable insights into our SAR campaign, in which we have a number of ongoing promising series with ever-increasing potency gains. So here we have a couple of CRCs from some of our initial screening hits, which have now developed into ongoing series and undergoing potency gains. So we plan to continue this SAR campaign, running the SyncroPatch alongside our Flipper thallium flux assay, as well as moving lead compounds through our cascade, which includes assays for target engagement, specificity, and proof of concept work with cell disease lines. As part of this, we've begun a collaboration with the Buchanan-Kelly Lab at the Dementia Research Institute, which is also at Cardiff University. And at the Buchanan-Kelly Lab, they've been investigating the effect of iPSC cortical neuron culture times on Parkinson's disease mutants. So we have begun replicating this MEA data on the SyncroPatch, measuring action potentials and IV traces. And we plan to use these models in our downstream proof of concept work. With a large and varied group of collaborators here at Cardiff, we are now looking for industrial partners to continue this work into a full-scale drug discovery project. And I believe with that, I conclude. So thank you very much for listening.
Wonderful. Thank you so much, Ewan.
Thank you.
We have had a few questions coming in. Ivy, there were some things about the transient transfection. Well, I was wondering what kind of was your efficiency, transfection efficiency with the MaxCyte that you had for the calcium channels. And then somebody else asked if there were any specific challenges in optimizing the transient transfection protocols for high throughput electrophysiology.
Yeah, there were a number of challenges. So we tried every other transfection method, including other electroporators, and it wasn't that we couldn't get the transfection efficiency high enough. It was that when we got the transfection efficiency high enough, we couldn't patch the cells.
Okay.
So the MaxCyte gave us the ability to really increase the transfection efficiency without losing cell health and cell quality. So we were still able to seal on it. So we ended up having to purchase a MaxCyte system, which we negotiated with them so that we could actually do those experiments. When we have 4 plasmids, our efficiency on the SyncroPatch is only at about 40%. So, which, at 384 is sufficient, but we are actually going to make some cell lines with some of the other subunits so that we don't have to transfect as many plasmids. What we found with Matt Trudeau has been working with us as well, and he did his HERG cells and transfected just a single plasmid, mutant and wild type, and that was 80%. So it scales with the number of plasmids. But if you can get up to 40% and be able to look at the different mutations, it makes a big difference because making a cell line for every one of those mutants just isn't viable. However, we are investing in making cell lines with the subunits to reduce the number of plasmids that we have to put in.
Okay. Another question for you, Ivy, whether you're planning to expand the SyncroPatch-based studies to include native or patient-derived cells. And if so, what would you expect from that? And what would be your concerns?
So we did do some patient-derived cells. The iPSC cardiomyocytes, I showed at the very end. I just don't have data on the newer mutations in the talk, but yes. So we're following, for the cardiomyocytes, we're following the protocols from Al George. And they do work pretty well. We're finding that for voltage clamp, generally the first time we put it on there, the protocols seem to work. For current clamp, it's requiring a little bit more optimization. And I think it's a little bit specific to the iPSCs because the resting potential of these cells is not actually as negative as adult cardiomyocyte should be. What we found is that when we use the low frequency clamp to hold the resting potential, we made the threshold too high. So it was very dependent to get the action potentials. We actually had to adjust that to what is actually native for those cells, or not use it. But that caused some problems with the analysis when you're doing it in the multi-well format. And so we just depolarized the cells a little bit and, down to, I think we were at -60 and we started getting really nice action potentials. Whereas down at -80, we didn't, I think that's the immaturity of the cells. We didn't see as many action potentials coming out.
Yeah, I mean, it was super interesting that you've also done some adult ventricular cardiomyocytes from mouse, right? On the SyncroPatch.
Yeah, and I will say that one literally worked on the first try. It wasn't super high. It needs to be optimized to get more wells successful. We've been doing all of this in the partial plate format. So we're sort of, testing things, and a lot of it was flyer experiments so that we could put in a grant. But the adult cardiomyocytes from the mouse, actually, it was the first, I had a student who was patching them and just took the cells that he wasn't patching, dropped them on the instrument, and it worked. So that was actually easier than the IPS.
Amazing. Not what I would have expected, but very nice to hear. Okay, I have a question for Ewan here from Marcus. Will you verify your hits recording proton currents in native lysosomes?
Yes, we do have that is part of our downstream pathway at the moment is to move some of these compounds to the solid supported membrane-based electrophysiology. So we've got an in-house surfer system, another nanion device. So yeah, that's definitely one of the next steps being to go in and validate with the proton currents, as well as looking at effects within cellular models as well, where pH is being assessed as well.
Mm-hmm. Would you do any like lysopatch with the SyncroPatch?
So it's something that we sort of keep an eye on. It's something maybe downstream that we're looking at.
Okay. Marcus also asks if there is a robust commercially available activator for TMEM 175 besides DCP-IP.
Yeah, so the activator space for TMEM 175 isn't quite as well. The activator and the inhibitor space aren't very rich, but the activator space is particularly poor. So you've got DCP-IP, which is a very strong activator. You've got zinc, which is comparatively weak, and then a few of the other known activators have, as far as I'm aware, questionable methods of action. So there's some stuff like SC79, where we're not sure if it directly modulates TMM175 or if it works through some kinases. So yeah, there's a bit difficult. There's no real good starting points in the literature, which is why we've gone straight into primary screening.
And do you think activators or inhibitors for TMEM 175 are going to be important for disease treatment?
I think the most straightforward part of 3 at the moment is with activators, because there's quite a bit of stuff going on at the moment with the prevalence of TMM175 in Parkinson's disease. And this is what we're currently working on with one of our collaborators. But I definitely think there's some scope also for inhibitors to be very valuable, either in drug treatments or in as a dual compound as well, just to further assess the function of the channel in these cellular models.
Okay, great. Thank you so much. I think in the interest of time, we are on exactly 1 hour. So I'm going to hand back to Jamar. I would like to just say thank you so much to Ewan and Ivy for these wonderful talks. If anyone has any questions, please, if anything comes up after the talk, you can email us or email Ewan or Ivy and they'll be able to answer your questions. So thank you so much, you guys.
I just wanted to remind you about the SyncroPatch grant, what Ali already mentioned. So just remember that the applications will open in September, but you can still register your interest now, and then you will get notified, and we will send you some reminders as the applications open and also as the deadline approaches. So feel free to register there, and also. If you are in Europe or coming around, we would like to invite you to the user meeting that will be happening in October from 28 to 29. We will have very nice talks, poster presentations, also some workshops, demos. So it would be very nice, and we would be very happy if you could join us here in Munich. Important to remind that you can join either online or in person. Yes, and I think that is all for today. So thank you for joining us, and we will see you at the next event. Thank you.